Hi, I'm Stella Lee from the Office of Gene Therapy and CBER's Office of Therapeutic Products, or OTP. I'll be presenting today's webinar with Dr. Emily Wires from the Office of Pharmacology and Toxicology, also in OTP. Today, we'll introduce you our recently published draft guidance titled Leveraging Prior Knowledge in the Development of Human Gene Therapy Products, Incorporating Genome Editing, which discusses considerations for leveraging prior knowledge for cell and gene therapy products. And we have the docket number on this slide. The main objectives of today's webinar is to provide you a high-level overview of this new draft guidance. We will first start by going over the scope of the draft guidance, then define key terms such as prior knowledge, public knowledge, and platform knowledge, and then spend most of the time explaining the types of information that can be leveraged by each discipline. Finally, we encourage you and our stakeholders to provide feedback on this draft guidance, and please send feedback on to the docket on FDA webpage, which is also listed on the previous slide. First, what is the scope of this draft guidance? It reflects FDA's current thinking on the types of prior knowledge that may be scientifically appropriate to leverage to advance product development. And it provides recommendation on leveraging CMC, non-clinical, bioinformatics, and clinical prior knowledge. We acknowledge that it primarily focuses on genome-edited or GE products, but most of the recommendations discussed in this draft guidance can apply to many cell and gene therapy or CGT products, including adeno-associated viral or AAV vectors, lipid nanoparticle or LNP-based products, as well as ex vivo modified cell-based gene therapies that do not incorporate genome editing. This draft guidance was released as part of PDUFA 7 commitment to publish a guidance on leveraging prior knowledge. Next, I'll define the key terms used in this draft guidance. First of all, prior knowledge is an umbrella term that refers to both public knowledge and platform knowledge. Public knowledge is scientific information that's based on widely accepted scientific principles that are typically long-standing. Platform knowledge is well understood and reproducible technologies that apply to product development that consists of the same or highly similar features across development programs. And examples of platform knowledge include developers' own data from previous products using the same or similar manufacturing platforms, publicly available platform knowledge, such as published scientific literature, as well as consortium and data sharing initiative knowledge. I would like to highlight and emphasize that the term platform used in this draft guidance is different from the terms platform technology or designated platform technology as defined under FDA's Platform Technology Designation Program. So what are the key principles for leveraging prior knowledge? Prior knowledge can be leveraged throughout a product's life cycle, and we will later discuss how you can submit prior knowledge information during clinical development and for licensure applications. The ability to leverage data does not require an approved product. It depends on multiple factors, which we will further discuss throughout this webinar, but it primarily depends on similarities in product design, cell source, manufacturing process, final formulation, key quality attributes, route of administration, dosing regimen, dose levels, and disease to be treated, etc. If differences exist, the degree of differences and the level of potential impact determine the extent to which prior knowledge can be leveraged from full data sets to partial information with supplementation. It is important to note that justification for the applicability of the leveraged data should always be provided. Lastly, we highly encourage you to discuss potential data leveraging opportunities with us as early as possible in your product development, such as during Interact and pre-IND meetings. We'll now discuss leveraging CMC information in the next few slides. The draft guidance provides examples of the type of CMC information that may be leveraged, including analytical methods and method qualification, validation data, lot release specification, stability data, comparability data, and process characterization and validation data. This is not an exhaustive list, and we welcome you to discuss potential leveraging opportunities with us for additional types of CMC information. Qualification and validation data of a platform analytical method may be leveraged across similar products, depending on the similarities between test articles. If differences potentially impact method performance, then product-specific verification may be needed. For example, for guide RNA purity assays, you may need to verify that method accuracy and precision are not affected by different guide RNA sequences. For lot release specification, you may leverage prior knowledge to justify which quality attributes to include in a lot release specification and suitable acceptance criteria for those quality attributes. For example, guide RNAs with a similar size structure, same manufacturing process and formulation, but a different spacer sequence, you may use the same lot release specification. You may also leverage prior knowledge to justify the need to include or exclude process-related residual testing using data from similar products with the platform manufacturing process. For example, testing residual ribonucleoprotein components for a cell-based GE product may be excluded from the release specification with prior RNP or ribonucleoprotein clearance data during culture for similar products. Please note that though additional considerations or justification may be needed to leverage prior knowledge for product-specific attributes. Another type of CMC information that may be leveraged is stability data. Stability data from similar products may support initiation of early phase clinical studies to justify stability of a product over the duration of the planned clinical trial. For example, stability data of an mRNA encoding one nuclease may support other mRNA's components encoding a different nuclease if they're stored in the same conditions and container closure. Prior knowledge of stability may also support commercial shelf life in addition to real-time stability data of primary product lots. For example, you may leverage stability data from clinical lots if those lots are representative, such as that they're manufactured with a commercial process, using the commercial formulation and the commercial container closure. Additionally, you may leverage in-use stability and delivery device compatibility data for similar products that have the same formulation and are administered using the same delivery device. For comparability data obtained from one product or component may support changes in other similar products or components. For example, data supporting a reagent change for one guide RNA may support the same reagent change when manufacturing other guide RNAs with similar structure. Last but not least, process characterization and validation data. Process characterization data between products using the same platform manufacturing process may be leveraged to determine process parameter criticality, define proven acceptable ranges of these parameters, and etc. Process performance qualification or PPQ data from one product may also be used to inform a new PPQ study design, for example, the number of PPQ runs needed, and or support process validation for a similar product using the same manufacturing process at the same manufacturing site. For example, PPQ study from one G product may support process validation of other product variants, incorporating different guide RNAs targeting different mutations in a single gene. This is my last CMC slide. I was only able to give you a high-level overview, so I encourage you to read the draft guidance document as it further provides additional details and examples of leveraging CMC information. With that, I will now hand it over to Emily, who will first talk about leveraging non-clinical information. Thank you.
Thank you, Stella. I will now talk about leveraging non-clinical and clinical information. For non-clinical information, the draft guidance recommends a stepwise approach that may be informed by non-clinical data from in vitro, in silico, and in vivo studies, including new approach methodologies, also known as NAMs. Considerations that should be taken into account for determining the scientific appropriateness of leveraging non-clinical data may include manufacturing process, formulation, intended therapeutic mechanism, route of administration, editor, dose levels, and dosing regimen. On the next few slides, I will discuss what this means specifically for ex vivo and in vivo genome-edited products. For ex vivo genome-edited products, non-clinical information that may be leveraged may include biological activity data associated with a specific genomic edit between products containing the same on-target genomic edits using the same editor. However, the biological activity of each editor and guide RNA combination should still be assessed, even if the on-target edit is the same. Additionally, product attributes that are independent of the genomic editing component may be leveraged across products. For example, the safety and activity data for the binding domain of a genome-edited CAR T-cell product may be leveraged, provided that the single-chain variable fragment component of the targeting domain and the cell type are identical between products. For in vivo genome-edited products, proof-of-concept data from a single animal model of a monogenetic disease may be leveraged to support scientific rationale. However, in vitro studies evaluating on-target editing efficiency in relevant human cells should be conducted for each editor in each guide RNA combination. Biodistribution data may be leveraged when delivery vectors are sufficiently similar, and differences are not expected to impact biodistribution profiles. For lipid nanoparticles, physiochemical properties such as lipid identity, ratio, polydispersity index, and zeta potential should be similar, and differences in cargo should not impact LNP physiochemical characteristics. For viral vectors, a similar capsid is required, and expression cassette differences should not significantly impact the gene expression profile. Additional non-clinical information that may be leveraged for in vivo genome-edited products may include general toxicology data if biodistribution profiles and product characteristics are sufficiently similar, as well as LNP genotoxicity data if all lipid components and formulations are sufficiently similar and not expected to alter the genotoxic profile. Other leveraging considerations include developmental and reproductive toxicology, also known as DART, and transgene-specific factors. For DART, biodistribution data showing no gonadal distribution may support foregoing DART studies. However, sponsors should consider a risk-based approach evaluating target tissue expression in reproductive organs, as well as biodistribution patterns. Early engagement with FDA is recommended to discuss these requirements. For transgene-specific factors, sponsors should address expression and safety patterns, codon optimization, and selection markers that may affect safety and activity when considering leveraging this information. Clinical experience may inform non-clinical programs. Clinical data from relevant products may inform safety risks and dose selection for a proposed trial. When doing so, sponsors should address the similarities and differences between products, the route of administration, and how prior dose levels and dosing regimens compare to those proposed in the new trial. For pediatric programs, clinical data from adult subjects may be leveraged in lieu of juvenile animal studies for determining pediatric starting dose levels. It is recommended that sponsors address the prospect of direct benefit and scientifically justify dose extrapolation from adult data based on similar disease characteristics and exposure-response relationships between adult and pediatric populations. Other non-clinical information that may be leveraged may include bioinformatics information. Next-generation sequencing and bioinformatics analysis are essential for assessing off-target editing risk and genomic integrity for every genome-edited drug product. FDA recently published a draft guidance, Safety Assessment of Genome Editing and Human Gene Therapy Products Using Next-Generation Sequencing, which provides detailed recommendations on how these assessments should be conducted. While product-specific off-target editing data generally cannot be transferred between products, a significant portion of the underlying methodology can be leveraged across different genome-edited products. Three major categories of bioinformatics information that could be leveraged are off-target assessment strategies and assay parameters, next-generation sequencing methods and sequencing strategies, and bioinformatics analysis tools and pipelines, which I will discuss in more detail in the next slide. Off-target assessment strategies and assay parameters. Off-target site nomination and confirmatory testing methods may be leveraged across genome-edited products using editors with an identical mechanism of action. Assay parameters such as the amount of starting material, concentrations of genomic editing components, and culture conditions may be leveraged when the intended on-target edit rates are similar between products. Next-generation sequencing methods. Sequencing strategies used for off-target site nomination, confirmatory testing, and next-generation sequencing-based chromosomal integrity analysis may be leveraged across products. For example, sequencing depth and specific sequence quality acceptance criteria established for one product may be applied to a similar assessment of other products. Bioinformatics analysis tools. A bioinformatics pipeline developed for a specific off-target assay may be leveraged across products assessed with that same off-target assay. However, it is important to note that certain product-specific parameters, such as the target sequence and mismatch tolerances, should be updated to match the corresponding product. Individual tools with broad utility, such as those used for sequence quality assessment or alignment, can be leveraged more widely across different next-generation sequencing datasets. Not all bioinformatics information may be leveraged. For example, the guide RNA sequence is one of the primary determinants of off-target editing risk. Therefore, off-target editing data cannot be transferred across products using different guide RNAs or sequence recognition components. On-target editing outcome data is specific to each genomic locus. Even if two products use identical editing mechanisms, the specific editing outcomes at different target sites cannot be directly transferred. Genomic integrity assessment results are influenced by both on and off-target editing activity. Because these results are tied to specific genomic locations, they may not be leveraged between products editing different genomic sites. Now moving on to clinical information. For leveraging clinical information, sponsors should consider how the characteristics of the platform technology may impact the clinical evaluation. Prior clinical data and information may inform one or more aspects of the proposed clinical study, which may include clinical trial design, clinical trial conduct or analysis, leveraging data from natural history studies and real-world experience, and leveraging data from long-term follow-up studies, which I will discuss in more detail in the next slide. Information that may inform aspects of the clinical trial design may include dose-limiting toxicity definitions, monitoring intervals and duration of monitoring, dose selection, and clinical pharmacology assessments, such as biodistribution, vector shedding, and drug-drug interactions. To inform trial conduct or analysis, sponsors should consider leveraging clinical data already collected. For the purpose of abbreviating the safety database, efficacy database, PL database, DDI liability, as appropriate, based on whether there are expected clinical differences, such as patient population factors that include disease setting and prior therapies or product-related characteristics. For leveraging data from natural history studies and real-world evidence, the guidance encourages sponsors who are planning to leverage natural history studies and real-world evidence to support future licensure to engage with FDA early in product development. This is important because FDA needs to evaluate whether the data is fit for purpose before it becomes deeply embedded in the development program. Fit for purpose is ultimately an FDA determination, and early engagement gives sponsors the opportunity to get that feedback before significant time and resources have been invested. Practically speaking, when a sponsor submits a proposal to incorporate real-world evidence into their development program, they should explicitly flag this in their cover letter, for example, by stating something like this. This submission contains a proposal to use real-world evidence. This language helps ensure that the submission is routed appropriately for internal review by FDA's real-world evidence team. This guidance also encourages external stakeholders to consider building shared databases that could support development programs across multiple sponsors, which could be particularly valuable in disease areas where patient populations are small. And finally, long-term follow-up monitoring is a critical component of gene therapy development and sponsors should refer to and follow current FDA guidance on long-term follow-up specific to their therapy type. However, FDA is committed to ensuring that monitoring requirements remain appropriately tailored to the evolving evidence base while avoiding unnecessary burden on patients and sponsors where the data supports doing so. Therefore, FDA remains open to considering modifications to certain aspects of long-term monitoring, such as the type of data collected, the frequency of assessments, or the duration of follow-up, on a case-by-case basis in rare circumstances where cumulative safety data and the overall risk-benefit profile may support a modified approach. Any proposed deviation from the current long-term follow-up guidance would need to be discussed with FDA and supported by a robust justification. Now that we've covered what can be leveraged, let me briefly address how to submit this information to FDA. Sponsors should provide a justification for the applicability of prior knowledge to the specific product. During the IND stage of development, information can be provided directly in the submission, or it may be cross-referenced to a previously submitted IND or master file. If the file being referenced has been submitted by someone other than the sponsor, a written statement of authorization to reference the information must be provided as per the CFR. For a biologics license application, while reference information that applies to drug substances, drug substance intermediates, and drug products may be leveraged, it must be submitted directly in the BLA per the CFR. To summarize, both public and platform knowledge can be leveraged across the product lifecycle, and this may include widely available or proprietary information. This applies to CMC, non-clinical, bioinformatics, and clinical disciplines. As a reminder, the ability to leverage depends on multiple factors, and we encourage early engagement with FDA to discuss leveraging during the early stages of product development. The comment period for this draft guidance closes September 1st, 2026. To submit feedback, we refer you to the docket information below. Lastly, we have provided the appropriate contact information on this slide for any additional information that may be needed. Thank you for your attention, and we look forward to your feedback.